it worked, but usually it takes really much time. So I've checked the server, and it's still written, the video recording is still, will be processed still. So that means today in the evening I will check it again. And so if in the cases it worked, of course, I will also upload videos. I will put them or somewhere so you will have an opportunity to check them again. So this is the first point I'd like to address. Second, there was an email with a request that we split this lecture in 2 days. Well, basically you're right, it would be a good idea, but there are several reasons why we cannot do it now. Because first of all, I need to consider the lectures from my colleagues, and we have already settled schedules, so I just cannot now shift everything and disturb my colleagues giving lectures. Secondly, I also have my schedule, which I cannot change so shortly. So the next 3 weeks, already every day is booked. That means I have some meetings, online meetings on that day, and I just cannot change it. So I hope that you understand it. So since some classes are a little bit too full with material, and for some people it's a bit stressy, well, what shall we do? I can just suggest that we split really the class in 3 parts, 1 hour each, so that every hour you have time to recover. And every hour we make a break. That means 3 hours, and maybe every time 10, 15 minutes long break in between. So it should work. Believe me, in some cases it's even worse. In our optometry program, I'm giving lectures in Kirchstetten, and sometimes we have lectures from 8 o'clock till 14 o'clock in the afternoon. So, which means just with 1 break in the, so 1 hour break in the afternoon, but it's like 5 lectures in 1 block. Same group, same person teaching. You can imagine how poor students feel, and also how we feel after this class. So what we have is not too bad still. Also, there was a question from Marina about West. I would not insist of you knowing all these fine things. It's of course great that you try to notice all even fine interactions between single proteins. I'm afraid even the story about interactions between WASP and ARP is a long story. It deserves definitely a single paper. So if you insist, we can still communicate via email, but I would say this is just going too deep. So this is definitely what I'm not going to ask in the exam. There are many things I'm not telling you. For example, also with NWEST, we have experience in our lab. We applied mechanical forces on cells, and we analyzed proteomic changes in the cells. And in fact, NWEST was the protein that was mostly overexpressed in the fibroblasts that experienced mechanical stress. So it was an interesting finding. But those are things that I'm not just telling you because they are just too fine and not to make it too much for you. Okay, these are 2 little organizational things. Now, let me overview briefly. We've spoken last week almost exclusively about actin and actin binding proteins. We've learned a couple of names, also actin-related proteins, ARP family. And also, I hope you still have a couple of names in your mind that you can relate to actin, like formine, profilin, cofilin, severin, those things I mentioned. One of the important points was the issue about phalloidine, that you remember is a toxic substance that stabilizes cytoskeleton. But of course, what we are talking about in the course, only like 40% of these gonna be asked in an exam. So of course, I'm aware that 60% you will forget, and in fact, it's normal that you will forget it. I'm just giving this additional stuff to make sure that after you've forgotten many things, still 40% maybe remains. And this is something that you will keep not for the exam only, but maybe for your whole life, and it will help you in some cases. As for homework today, as you maybe noticed, the homeworks we have are actually not for me. They are contributing to your knowledge, to your understanding. And also today's homework is not going to be challenging or tricky. Later I will give some more challenging homeworks, but today I suggest following. Let us consider as a homework the following activity, which is of didactic importance. That is, let us make a table at home where you will summarize and put in a very structured form information concerning 3 types of cytoskeleton. So I suggest you to make 3 columns in your, well, actually 4 columns in your table. The first column will be property. The second column will be microfilaments, actin filaments. Third column will be microtubules, and the last column will be intermediate filaments. So we will have property, and then we have 3 different groups of cytoskeleton molecules. And what you can do, and it would be really good for your memorizing stuff and preparing for the exam, is to start filling this table and comparing between 3 different types of cytoskeleton, so that in the end you have a nice picture of the of the whole cytoskeleton. And then for the exam preparation, just look in this table and 5 minutes long, and you say, oh, I got it, that's really nice. So here's the question to repeat about columns. It's up to you. I would suggest to do it. First column will be certain feature, certain property. Second column will be actin cytoskeleton. And next column will be tubulin cytoskeleton. And finally, the last column will be intermediate filament cytoskeleton. And I think it would be a good homework activity for you, even starting from today. Let me suggest some possible features that you can compare concerning different microtubular types. For example, you can put representatives in one, or like building blocks in one row. So in the case of microfilaments, definitely these are actins and partially are ARPs, right? Actin-related proteins. In the case of microtubules, there will be alpha and beta tubulin. Then you can put another row which will be about shape and polarity. And in this case, we mentioned that actin blocks are almost spherical, well, strictly speaking, conical, and that they have polarity. You just say have plus and minus ends. And as we'll see today, microtubules have also plus and minus ends, but intermediate elements, they don't. Another feature you can have in your table is ATP or GTP dependence. So in the case of actin cytoskeleton, we already can put there. Actin polymerization is not strictly dependent on ATP, right? But it is controlled by ATP, right? Then you can put some feature like two main functions in a cell, three main functions. And let us overview about actin. For example, we've learned that actin can exert pulling forces together with myosin. This is a muscle contraction. It can also make gentle pushing forces, this branched actin network, together with ARP, they can push membrane, making protrusions. We also learned something mysterious about water conducting role of actin, what we observed in microvilli, and so on. There are many functions, so you can choose any which you like more and make a note. Then we can also make additional role associated motors, associated motor proteins. And I think in a case of actin, you already know what I mean. We're talking about myosin. So there are different myosin types, and I think we haven't spoken about this much, but it is myosin that is the main partner in exerting forces. Well, there is then hypothetic partner as well, ectoclampin, but nobody has found it so far, so that's why we just leave it aside. And maybe, if you wish, some of you are very motivated to learn more from this course, you can also maybe include raw binding proteins. So in our case, we have spoken about actin binding proteins. We mentioned many names. I have given you around 15 different names. If you just remember 2, 3, 4, 5, whatever, that's already fine. More you will not need. Well, that's my suggestion. So I hope you will like it, because indeed it will help you to organize your knowledge. I'm not requesting that you send your tables to me. So this is the activity you are going to do for yourself, by yourself. And if you wish, of course, I would be happy if you send such a table to me, just to check if everything's correct, or maybe have some suggestions, or we can discuss about some terms and columns. Of course, surely. So. But it's up to you. So that was about homework. So maybe later, if somebody have another idea about what could else appear in our table can be long actually. Please let me know. We can include this feature as well, and or in fact, you can do it for yourself. Or maybe some people would like to specify what kind of forces applied, or something about size and thickness. Of course, nice, why not? So, again, let me mention the plan for today. We will start with microtubules. In between, we will talk about some motors related to microtubules. Then we'll speak about intermediate elements. And then we will talk about tensegrity concept and its main statements, pros and contras. And well, maybe finalizing, we will try to overview some fundamental principles of cellular motility. So locomotion and motion events in the cell, since it's a biophysics course. Today I'm going to mention some additional terms such as dynamic instability, catastrophe, rescue, catastrophins, and so that's why it would be useful to take some notes. So now we're going to start, and just very last remark. There were some feedbacks from a couple of people that we are sometimes too fast. Maybe you are right. Actually, I try just to make it as always, so trying to keep the pace more or less uniform, not uniform, but making some unimportant things more fast, and the things that are really important for you. Usually I am slowing down and I'm repeating it, and I think it should work. The problem is that if I'm getting slow there, then in the evaluation later, there will be definitely half of people who will mention that it was too slow. That happened to me once in my chemistry course, where the notes evaluations were quite good, but half of people said it was too fast, and another half is that it was too slow. Well, so let's see. Of course, you always have opportunity to stop me and to ask something if it was really too fast, and you know that I am responding to that. Our next cytoskeletal component is our microtubules. And I think we keep our table in our mind, so that's why we have now some inner structure in the material. So we're talking about maybe mechanical properties, monomers, functions, binding proteins, and so on. And meanwhile, you can also fill this table, or maybe later at home. So microtubules are considered as the stiffest element of cytoskeleton. This is the only element of cytoskeleton that can truly be regarded as stiff. So we know that also actin can gently push membranes, but in fact actin filaments are not so stiff, but microtubules they are. And I think if you just inspect the picture, you understand why it is so. So seen mechanically, biomechanically, these are hollow tubes, so with lumen inside, and in fact such a structure can indeed withstand mechanical forces with high efficiency. And so we'll see later, microtubules in the cell they're performing many functions. They can pull, they can push, and they can guide certain molecules and some other functions. What we also see from this slide is like actin filaments, microtubules, they also have plus and minus ends. And like in the case of actins, the plus end is the fast, rapidly growing end, and minus end is slowly growing end. So we're talking about certain intrinsic functional polarity of microtubules, like actin filaments. They both have functional polarity, structural polarity. The main building block of microtubules are dimers of tubulin and alpha and beta tubulin. In fact, alpha tubulin and beta tubulin are two different proteins. They are quite similar. We say they are homologous. That means they are quite similar in their structure, and they have same origin. They are copies of the same gene, actually. Actually, I should say their genes are originating from the same gene. And so they are quite similar in their primary sequence. And interestingly, they can bind very strongly to each other with high affinity, with high bending constant, forming dimers. So we will consider this alpha-beta dimer as the building block of tubulin. Alpha-beta tubulin dimer is a main building block of tubulins. You also have noticed, I think, that red written is that it is a GTP-dependent process, guanosine triphosphate. Interestingly, right, we have actin polymerization that was controlled by by ATP. And now we have a certain differentiation in the cell, right? So we have microtubular processes, they will be greatly controlled by GDP, another nucleotide. Quite interesting, and I think our anthropocentric explanation of that will be that the cell kind of wanted to control actin-related processes separately from those microtubular or with microtubules. So if we play with ATP concentration, we can orchestrate something related to actin, and in fact, independent of that, we can also play with GDP concentration in the cell, and this will influence another compartment, another player, microtubules. So this will be my anthropocentric explanation of this. So at least this is something that we can that's worthy to note. GDP dependence. Also, I think not needed to explain here when there are some size data concerning microtubules. They are rigid, they are large, the largest, the thickest structure in the cytoskeleton. And good is that you see those pictures that appear in your mind, in your visual brain will maybe soak certain pictures, and they will help you to order information better. The last remark I'd like to make concerning microtubules in this slide is their distribution in the cell. Let us recall something about actin distribution. By the way, you can also put it in your table if you wish. Actin was omnipresent in the cell with main concentrations with the highest density located in microvilli, in stress fibers, close to the nucleus, even inside the nucleus. So these are places of highest actin concentration, but it was everywhere. Oh, I've forgotten to mention anchoring junctions, which will appear later in this course. So the places where cells attach to the neighbors or to the substrate, they will be all accompanied by high actin concentration. So, but don't worry, we'll talk about this later. There are good pictures about that. Now let's see what we know about distribution of microtubules. So the main pattern we see for microtubules is this star-like pattern. They have all common center, which we will call microtubule organizing center. It has also abbreviation. It's called MTOC, microtubule organizing center. And most of them, in most of cases, they will originate from there. We will, in a short time, we'll talk about why. And this is what you find in many handbooks. Let me correct some handbooks because recent papers show that it's not always true. In some cases, we observe tubulins to be arranged in very different structures, architectures, maybe close to membranes sometimes, or also in the parallel arrays. So that's why if we keep it correct, we should say in many cases we observe this typical star-like architecture, but there are exceptions. Well, I think this is what I wanted to mention concerning these pictures. So here are microscopic photos of microtubules. And as I already mentioned, this warning sign signalizes that you should that you can absorb a lot from this slide, that it's important for exam. So I think that was it. So you notice that this slide was important, and we spent a lot of time with that, and I talked a lot, and I repeated a lot, and I think you understand what it means. And some others I will just kind of make very quickly, and I think you also know what it means. So here are just some fine details concerning the structure of microtubules. As mentioned before, our main building block is the dimer of alpha and beta tubulin. You also can see that even their structure is quite similar. So we have certain special arrangement of alpha helices and little elements of beta folding, but almost exclusively alpha helical structures. This yellow labeled spot here is the GTP binding site of microtubules. And you could see also interestingly, as two monomers, alpha and beta tubulin, bind together, one of the pockets for GTP binding will be closed. And this is what makes this process also interesting. So GTP facilitates binding between them, and then it serves as a glue, and then it can not exchange to the environment anymore. And now the two alpha and beta subunits are really stick together, and they are glued together by GTP. And that's quite a strong connection. That's why we say mostly they don't dissociate in the cell. So here are some protofilaments, but it's not so important. Some people also find something like a seam, like connection plates between some weak plates in microtubules, but those things are not important. These are just for fun. Now, let me again initiate some other important topic, and it is about microtubule stability and polymerization. And I think as you inspect the lower part of this picture, you already could guess what I'm going to say at the beginning. And this is that if we start with monomer and single dimer, and we want to make a microtubule out of it by polymerization, again, the initial steps of this process are very slow and unfavorable, right? Exactly the same what we saw in the case of actin. The first steps are really problematic. They occur each step with very low probability, and that's why without any nucleation helpers, some proteins or some structures that will help, that would support this process, this will not occur maybe. So it's really very slow process here. And then after we've reached certain critical mass, the polymerization occurs spontaneously, and it's favorable. And this is what you see as the thickness of these arrows. So the thickness specifies the rate constant here, right? So the rate constants are higher, getting higher as we proceed with polymerization. So here. Okay, that means already I think we established some bridge between actin and um, microtubule, tubulin. Oops. Okay, it just crashed. I need to start it again. Just a moment. So, all right, I just need to put your chat window to another monitor so that I can see your comments live. And unfortunately, also our recording row. So I need to start it also again. Well, let's see, maybe it has recorded something already, and I will later just need to join the lost fragments. Okay, uh, so I think nothing really new. We saw many analogies to actin, but now slowly we will see, together with similarities, I will show some very unique features of microtubules, and we will spend a couple of minutes with that. So this is first of all, again, some sort of what we saw in actin before. This seed, the slow step, nucleation step, and after it happens, we now have opportunity for microtubule to grow faster. So that's just a repetition. And of course, it shows itself on the level of the curve of polymerization. If we have no nucleator, no helper for tubulin, then it takes really for a very long time. You see again very long lag phase, then we have exponential phase, and the last where we have saturation, right, where we have equilibrium. And in the case, we have a nucleator, and this is mostly the case in the cell. So this scenario is realized. And then the polymerization occurs quite rapidly, quite quickly. So the slope of the curve tells us that it's really quick process. And in addition, it's controlled by bivalent cations like magnesium, calcium, and monovalent also lithium. So again, we can compare now critical concentration of tubulin and actin. I think some of you remember the critical concentration of actin was 1 microgram, or even less, 0.1 microgram. And in the case of tubulin, we have higher critical concentration, a couple of orders higher. Well, you don't need to do these numbers for the exam, actually. I'm just giving it for fun, just that we can compare better. And now something more interesting appears, and this is where we are going to meet a phenomenon that is called dynamic instability. And I want you to notice this, please. Dynamic instability. This is a special term that is used to describe tubulin dynamics. As the name suggests, tubulin or microtubuli, they are dynamically unstable. And this is a bit paradoxical because on one hand, I said they are, very stiff structures, thick structures, very imposant structures, like looking stable actually, but in reality, they are not. And again, let us recall what we talked about actin. I asked you what could be the reason for the cell to use spherical building blocks to make mechanically strong structure. And there were some interesting hypotheses, but I believe that one of the main reasons is that there are some reasons to make the structure rapidly appearing and disappearing, very, very labile. And it was true for actin, and it's also true for tubulin. But in the case of actin, we have not observed this phenomenon of dynamic instability. What I mean, let me describe. In the case of actin, we mostly saw that we have plus and minus ends, and if we have a favorable situation, then both will grow, one end slowly, another end faster, and everything is fine. If we make concentration different, we will play with critical concentration, then we can see one end is growing faster, another end is not growing at all, and or even decomposed, and this is what we call treadmilling. But everything was sort of linear and predictable. But in the case of tubulin, the situation is different. The dynamic instability of tubulin also is described by another term, and I think this term will reflect this feeling of the difference between them. I will write this term. So they call it catastrophe. So not with Y in the end, but with E in the end, catastrophe. I think you see it's very strong words. So it means very dramatic change takes place. And indeed, what tubulin undergoes, or microtubuli undergo in the cell, is called catastrophe, and it happens relatively quickly. So in an average cell, the frequency of this event is 0.014 per second. So here's the number. It happens quite often, so with high frequency. Let me describe now what it means. You observe a microtubule, and it's a happy microtubule that just grows, and it grows slowly, and it's getting larger, and everybody's happy. And suddenly, and it's a totally random stochastic process, it starts to decompose, to depolymerize with very high speed of approximately 1,000 dimers per second. Which would correspond to approximately 0.5 micrometer per second. That's really fast. I mean, if you observe a microtubule and it suddenly initiates this catastrophe, you will really see it under microscope. So you will say, oh, wow, what happened? So it's really something like an explosion, actually. They very rapidly lose their structure and decompose. Honestly, we don't know why and how it happens. So it's a thermodynamically controlled process. So it's just a random event. It's a stochastic event. And this is an intrinsic feature of tubulin to initiate such catastrophe. And then after a couple of seconds, it's over. And again, this filament will start growing again slowly. And again, after a certain time, it will say, oh, it will decompose again, a couple of seconds long, and again, it will grow again. So this is the game acting, oh, sorry, the game tubulin filaments, the tubulin microtubules, play all the time. So everything is okay. So I now introduced one peculiarity feature of tubulin dynamics, which is called catastrophe. This is the very rapid decomposing event, and it happens randomly without any proteins, just by itself, without any helping proteins. Little update to this. Sometimes this catastrophe event, it starts, and you see, oh, it begins. And then suddenly it stops. So it does not go to the full extent. And in this case, we are talking about another special term. It is called rescue. The rescue event. Again, it's a sort of dicing in the cell. Both catastrophe and rescue events are random. It is like you are dicing two times. So first you dice, and or you throw a coin, and if the coin is in this side, you start catastrophe. And if you throw it one more time, again, depending on which side the coin you have, either catastrophe proceeds or it will be stopped by rescue. And this is really mysterious, but this is what we have in the cell. And these are two terms that good describe the protein instability or dynamic instability in the cell. So now let me show this more clearly. So these are some events related to dynamic instability. So here we have the microtubule organizing center, MTOC, that I mentioned before. This is a nucleator in the cell, and many microtubules in the cell, they grow from this center. It has certain capacity, so that means it has some place, but it cannot allow, I guess, more than 100, something like this, microtubules to originate from this center. It just has no any place anymore. So that's why it has limiting capacity. And in order to describe these catastrophe and rescue events, the methods of the deterministic chaos are applied. Very beautiful models, and here I'd like to recommend to all of you who are interested in dynamic modeling, in chaos modeling, chaotic events. There is a famous mathematician, Stephen Strogatz, who published many books about mathematics of chaos, fractals, fractal events, and chaotic events. and how mathematics tries to approach a random and stochastic event. So really nice. Oh, so Caroline has written that there is a problem with sound. Okay, thank you for that. Oh, that now you can hear me again. Fine. Yeah, thank you very much. First of all, it's very important because sometimes I'm just asking myself, so did somebody hear me? And secondly, well, okay, it's just internet connection, so sometimes we lose signal, and well, uh, but it's not too bad, you know. Later you can anytime ask me, and so Caroline is asking if I can repeat what I've talked about in the last 5 minutes. In order to not to make it boring for the rest of the group, let me just repeat the uh, three main terms I mentioned here, and those are dynamic instability, catastrophe, which you find also in the chat window, and rescue. So microtubules undergo catastrophe, and let me now show actually catastrophe on our picture so that everybody can see it. This is the happy growing microtubule, you see? And then this is the moment when, for some reason, nobody knows why, this random stochastic catastrophe event occurs, very rapid depolymerization, and then it starts growing again, and then it's somewhat like again catastrophe happens because it just happens. And then this tube, microtubule, start growing again, and again catastrophe is coming. And here one more growth, and here catastrophe again. So I think in this case, in between upon its growth, maybe, oh, here you can see the rescuing event, short rescuing event. So catastrophe started, then rescue happens, so it starts growing again, and then next catastrophe occurred again. Rescuing is another random event that occurs after catastrophe occurs. So it's like it can happen that catastrophe will be broken, cancelled by so-called rescue. So this is a term, and it defines a situation when a microtubule starts losing its length and then say, "Oh no, let me grow again." And this is what we call rescue. So as for equations, I think you don't need to inspect them very thoroughly. If you like, it could be a good exercise for you in terms of rate constants, but they are not very informative. They are just telling how we try to model these chaos dynamics. And again, I would strongly recommend really very brilliant videos and books from Steven Strogat. He explains chaos mathematics in a very understandable way. So here, I just don't want to repeat his ideas because he does it in a really brilliant way. He is a perfect lecturer, and I just can recommend it to you. If you want, of course, I'm not asking about Steven, right? Not asking about his lectures in exam, right? So just bonus information for you. What I wanted to share with you. I just found it really fascinating what he explains. Fine, we almost So let me summarize now briefly. I promised that in our class we make some breaks to make to keep you fresh, to keep your attention like alert. Let me summarize briefly what I've spoken about before. We mentioned some key features of microtubules. We try to understand them in a comparison with actin. We could establish several similarities and dissimilarities between actin and tubulin. And I think many of us already can put something in our table where we compare their properties. To finalize this chapter and to establish a bridge to the next one after the break, let me mention one more name, one more protein name that participate in this dynamic instability. I will write it down. You will see it easy to memorize. Catastrophins. So these are proteins. This is a protein family that contribute, that participate in those random events. I mentioned before that catastrophe occurs in the cell with pretty high frequency, so 0.014 times per second. But, so this is the background level. There is a special pool of proteins, group of proteins called catastrophins that interfere this process or control this process or influence this process. There are catastrophins that increase the probability of catastrophe to happen. These are catastrophins that actually do not allow microtubule to grow. If they are present, it will always experience catastrophes and cannot grow at that place. There are another catastrophins, but in spite of the name, they are helping tubulin to grow, microtubule to grow. So they are inhibiting catastrophes and acting rescuing. But in both cases, they have same names. They are called catastrophins. So these are proteins that, in addition, allow cells to fine-tune the process of the random process of dynamic instability. So I think that's enough for now. I hope you are still not very tired. We did not so much, but in order to keep your attention high for the next section where we talk about Alzheimer's disease and tubulin binding proteins, I'd like now to announce our short coffee break, 10 minutes long, and then for the next hour, we will stay active and we will talk about interesting events happening with microtubules. And you'll see it's quite interesting, quite fascinating what's going on there. So now for 10 minutes, I'm switching my micro off and we see us in 10 minutes. Enjoy your coffee break, right? Maybe drink some coffee or take some fresh air. So see you. So, sorry, by the way, I spelled Steven's name with a mistake. So actually, I've forgotten T. So now I have corrected it. So on my slide, but in your script, it can be still wrongly written. So it's Strogatz with T. So please correct if you have it in your hands. So, and now let us continue. And in the current time, we are going to talk about tubulin binding proteins, microtubule stability in the cell, Alzheimer's disease, role of microtubules in our consciousness. And then I think we slowly can move towards, oh, actually I will show some cancer-related example about them. And then we will move towards inhibited filaments. I think this will happen in the last chapter. Let's see. As for microtubule binding proteins, they actually are terms not microtubule binding, but the official name is called microtubule associated proteins, MAPs. MAPs, microtubule associated proteins. And there are lots of pathways that is related to MAPs, so many signaling pathways, which we will learn in a final part of this course. They have microtubules as one of the main targets, as one of the main objects. For example, MAP key signaling pathways where cellular kinases pass the signal into the cell. It will, in many cases, it will end or it will aim to the microtubules. So obviously, microtubules are very important players in the cell. And why they're so important, what their roles are in the cell, we will talk after this slide. Now let us see, we know now something about the structure of microtubules, the main building blocks of alpha and beta tubulin, and actually that's it. So this is the core of the microtubule. This is what they contain. But at the same time, it is just a part of the story because there is a huge group of proteins that modify microtubule functioning and microtubule properties. And let me now mention some of them. One of them we've already well noticed and met. This is catastrophine, or even family of catastrophines that can stabilize or destabilize microtubules, right? Let me mention another name, which is also very much, telling about function. This is katanin, like Japanese sword, katana, right? So what it does, it cuts, right? And it's very good at that. And that's why it's very easy to remember that there is a special protein called katanin, and its role is severing, cutting microtubules if it's needed. So it's a controlled cutting of microtubules in the cell. Katanin, katana. We also see here that this is a GDP-dependent process, and here I will not focus too much because GDP regulates tubulin polymerization in a similar way how ADP does. So actually it is about changing changing rate constants of the polymerization reaction, which allows for more control for the cell over tubulin polymerization. One name is not mentioned here. Maybe I should add it. These are so-called stat means. In the last years, stat means, I will write this name in our chat window. In the last couple of years, stat means they become very important because some researchers consider them as potential drugs that can help against Alzheimer's disease. And also, obviously, that means they participate in many signaling events in the cell. So this is pharmacological industry focal point at the moment. Statins are proteins that mostly slow down tubulin polymerization. So mainly they prevent polymerization they prevent growth of microtubules. So this is how I define their main role actually. Prevent microtubule growth or slow down microtubule growth. There are a couple of more names, but I'm a little bit, okay, I will still mention some of them just to give a picture for you. Let me also mention some motors, and it's very interesting that molecular motors, they are also in this list. They are defined as microtubule associated proteins. And here I'd like already to mention these two important molecules. They are called dynein and kinesin. We will see later what they are, what they do. I can write. Just when I'm writing spontaneously, sometimes I'm making some spelling mistakes. So that's why do not rely too much on my spelling. Later in the slides, you will find definitely these names as well. So these are two motor proteins that are also working together, necessarily working together with microtubules. And by the way, we can also fill our table now. So the actin has myosin as molecular motor, and microtubules have kinesin and dynein as molecular motors. About the role of tau protein, which is shown here, I will tell you a little bit later. I will come back to it. And now I just want to draw your attention to the fact that this figure contains subunits of different colors. So then you might ask yourself, why? I mentioned before, these are just alpha and beta tubulins. Why do I use different colors here? What do I want to say with it? Let me explain. One interesting feature of microtubules is that they are very actively modified post-translationally. So post-translational modification. That means already synthesized proteins, and even later, as a part of the microtubule, they are actively modified by many proteins. What do I mean modified? One possible modification is that certain phosphor acid residues will be added to microtubule. It could be glycosylation. So some carbohydrates may be added to the molecule. It could be polyglutamate residues will be added to it. So there are over 20 different modification opportunities, and I found it quite mysterious because seemingly the cell makes lots of efforts to modify single tubulin monomers inside of microtubule, but nobody knows why it takes so much effort and what's the meaning of this, what's the role of this. One funny thing I want to mention here is in our course, Cellular and Molecular Biophysics, we talked about proteins. In the first part, we talked about proteins, and we noticed that the primary structure for protein is linear, so it's a linear polymer, right? So, and only after it starts secondary and tertiary transformations, it gets certain shape and blah, blah. Now something interesting was coming. In most handbooks, you will find that our proteins in the cell are not branched, right? They are linear. And this is what our current state of knowledge is. Proteins in the cell are linear polymers. There are no branches. They are not capable of branching. And now the funny thing is coming, and it is, there is at least one exception. And the exception is microtubular subunits. Upon modification of microtubules, some branched proteinous structures appear there. And this is a very unique case, actually. Here, some branching occurs. So let me now make some ugly drawing that should illustrate this. So there are several proteins that create branched protein trees on the surface of microtubules. So in fact, if you observe microtubule in the cell, it will not be a naked microtubule, but it will be covered by branched protein chains. Again, very strange, quite mysterious. A couple of years ago, there was a paper in Nature about this phenomenon, but still most of biophysicists and cell biologists say, oh, no idea. As for other proteins, clasp systems, and ADP kinases, please let me now skip it a bit because I don't want to overload you with things. If they appear later in the signaling pathways, let us consider them. But mostly these proteins are, sometimes I talk more about them, sometimes not. It's just an additional thing. I hope you forgive me if I just skip them for now. And it's enough for us if we notice catastrophins, catenins, and motors and tau proteins. So that would be enough. Now, as we've learned a little bit about the machinery that accompanies microtubule functioning in the cell, let me briefly overview the spectrum of tubulin and microtubule functions in the cell. And microtubules are also very important. Look what we have here. These are absolutely critical events. Cell division. In particular, it is about separation of chromosomes. And if it would be the only function of microtubules, that would be totally enough. Just think about what happens if microtubules fail in doing the proper chromosome separation upon cell division. What if one cell has one chromosome more or one chromosome less, you know what happens? We have trisomy, we have Down syndrome, we have Kreutzfeldt-Jakob, no, how's it called? Okay, there are lots of genetic diseases related to an absence or one chromosome more, and that's absolutely critical function, right? That's why no wonder that we have many additional proteins that help microtubules to perform their functions, because we cannot rely on naked microtubules. You know why? Imagine you have a microtubule that is now pulling our chromosome into one of the daughter cells, and at that moment, this microtubule undergoes catastrophe. So it says, okay, I'm off. And so it will be also a catastrophe for us, for the dividing cell. So that's why the cell needs to apply additional mechanisms to gain some risk management, to perform risk management, you know, to get more control. Next important function I want to mention, there are about 10 of them, but let me mention the 3 most important. Axonal transport. What does it mean? Let's consider a typical neuron in our brain. Not in the cortex, but maybe the one that is in a thalamus or hypothalamus. Such neurons, they are so long sometimes. Or let's take a ganglion cells in our retina. They are so long. They have long axons that are 10 to 20 centimeters long, guiding signals from the brain to the periphery. In order to perform as a single cell, you need very efficient transport mechanisms that would transport substances from one place to another. Consider a neuron which needs a neurotransmitter to be continuously restored in a synaptic cleft, in a terminals, and for that you need transport. Transport is nothing without pathways, without let's say roads, and the microtubules are the roads that the cell uses to transport molecules. So please let us associate one more important function of microtubules. They are highways of the cell. They are Autobahns, like we could say in German. It's highways that are straight, that are broad, and these highways are used to transport substances from one place of the cell to another. If this concept is new for you, you might ask yourself how it occurs. We will talk about this a bit later. Now I want just to mention that there are basically 2 opportunities. You remember, cells are, microtubules are tubes, so they have a lumen inside. So in fact, it has been shown that in some cases, some compounds can pass microtubules through this opening. But this is rather an exotic and non-typical way to transport substances. Most of substances are transported along the microtubule surface. So I have a microtubule here, and there are motor proteins that carry cargoes in one or another direction, but they use microtubules as mechanical support, as roads that guide their motion. So cell division, chromosome separation in particular, axonal transport, and especially true it is for neurons, of course, in their axons. And finally, cilia and flagella. Well, that's definitely also one of the most critical functions of human body. Think about places where we have cilia and flagella. Just take a couple of seconds. So where in our body we have cilia and flagella? Which cells? So if you are talking about intestinal cells, then you are wrong because these are microvilli. Nasal cavity is good, so respiratory pathways, right? Respiratory epithelium, right? And this is also not true for the nose only. It's true about our bronchial pathways. They're all full with cells that having these ciliar structures. And of course, spermatozoids, because without motility mechanisms, without their locomotion, no natural fertilization can occur, because maybe from the developmental biology, you know that in order to meet oocyte, spermatozoid must go through quite a long way. It's several centimeters long, which is a huge distance. It's a marathon distance for a single cell. So they need to go there, because without that, they would not meet oocyte and no fertilization will occur. And that's why without functioning microtubules, it will not happen. And the only option that remains is artificial fertilization. And since in both the cellular types, in respiratory epithelium and in spermatozoids, we have same mechanism, and no wonder that people who have some gene mutation related to microtubules or dynein, they have two problems at once then. They have problems with their respiratory pathways, and they have problems with fertility. So if they are males, they are mostly non-fertile, and the only reason for that is the mutation or improper functioning of microtubules. Here I want to mention some, just on the side, you know, the mechanism of our protection against most of microbes and also viruses and coronavirus as well. In fact, in our respiratory epithelium, we have continuous flow of slime, of mucus, and it is in constant motion. And it's a very important mechanical way of protecting our body. We are exposed to bacteria and viruses all the time, and we have in fact many our own viruses and bacteria living in the nasal cavity and in the mouth, but they are our own, and mostly they cannot invade because of slime motion, because of mucus motion. It is like they try to enter the cell, but the mucus protecting layer drifts them away, and that's why we stay healthy. But there's a constant attack of our body, even from our own microflora. But at the moment, we have colds, or if it's cold outside, at low environmental temperature, the cells, the beating pattern slows down, so they cannot do it too fast. If it's cold, they are doing it slowly. That means the mucus speed slows down, and now viruses and bacteria have enough time to enter the cells. This is what also explains why in winter time, especially, we are so sensitive, so prone to colds, to influenza, to coronavirus, to many other pathogens, because this mechanical flow mechanism in our respiratory epithelium works slowly. So, and maybe you knew this, but I wanted to mention this fascinating mechanism that protects us from pathogens. So let's now check some figures. I'm showing here spermatozoid. Here is the fragment of these beating cells in our respiratory epithelium. So they continuously doing this. it's a wipe, like a wipe eye chair, yeah. So it's like wipe motion. And here, this is the figure I wanted to necessarily to show you. This is something that you don't see often. You see, this is a microtubule here, and this is another one. And what you see here, microtubules, these single dots you may see in the figure are single tubulin polymers. And as you notice, some of them are dark in color, some of them are gray, and there are different shades of gray here. And this is exactly because of the reason I mentioned before. Tubulin subunits, they can be post-translationally modified, acetylated, phosphorylated, glycosylated, polyadenylated, and so on. And that's why so complicated pattern appears. So it's like a hidden code you find written along microtube. It's a little bit like on a hard disk, actually, where magnetic domains encode information along a microtube. We have lots of differently modified tubulin dimers. That's a code, that's a language that we still don't understand. Well, it might sound too esoterically for us, but let me show some more practical example. And in this case, I'd like to mention tau protein, as I promised before. Tau protein. Sometimes it's just written with Greek letter tau, right? So you know this letter. Or sometimes just like this. I promised that I will tell about this protein, and for the good reason. This is a protein that binds to existing microtubules from the side. So there is a microtubule, and from the side there are many proteins that bind to it from the side. You remember still about this branching protein trees. So one of these proteins is a tau protein, and the tau protein can stabilize microtubules to prevent their catastrophe. And I think you understand how critical it can be to stop catastrophe and prevent destruction of microtubules. So here's the example. I know that some slides you don't have in your script. Some, maybe this you have, but I'm showing some that you don't have. This is for many reasons. Many of them, they just repeat each other. One research group in Germany, the research group guided by Professor Andrea Robitzky in Leipzig. They are our research colleagues, research partners in some projects. They studied the role of tau protein in pathology of Alzheimer's disease. It's also partially their findings, what I'm telling now. So it is known that tau protein stabilizes microtubules, but for that, tau protein needs to be mildly phosphorylated. That means it needs to have, I guess, 3 phosphor acid residues. So it's kind of activation, but not overactivation. So 3 phosphor acid residues. And what has been found also by the group from Professor Robitzky is that tau proteins are getting over phosphorylated in the Alzheimer's disease neurons. So this additional phosphorylation, which occurs 4 times, 5 times, or 6 times, makes tau protein not functionable, And so there will be clumps, aggregates of tau proteins that appear in the cell, and it cannot stabilize microtubules anymore, and they degrade. And what we see is, first of all, we see aggregates of tau proteins in the diseased neurons, and secondly, we also see that microtubules are not stabilized anymore, and they fall apart, and this is, of course, devastating for neurons. That's absolutely deathful for neuron functioning. Neurons cannot pass signals, their structure is collapsing, transportation mechanisms are broken, and what we see is Alzheimer's disease. Honestly, this is just one of many hypotheses that try to explain the mechanism of Alzheimer's disease. Of course, the other one is related to amyloid plaques and, protein presenilin. At the moment, there are 8 different, totally different hypotheses explaining Alzheimer's disease. But this is one of them, and there are also some proofs that in many cases it works really like this. So here's just one more picture that illustrates this process. Now, let me tell you something that might be interesting for all of you, because Alzheimer's disease is something that can affect any of us, right? Sooner or later. In the hereditary cases, in the case of a genetic nature, the disease occurs mostly, can appear with 50, with 55, with 60 years. And in the case of idiopathic origin, that means if we don't know some random processes, most of people get sick with 70, 75, 80, and this affects many people, in fact. And I think that's why this is a topic that is interesting for everybody, because first, it can affect any of us. Second, there is no remedy. We can only slow it down, but we cannot cure Alzheimer's disease. And that's why let me share 2 ideas with you that might be interesting for you. First idea is Alzheimer's disease maybe is not one disease. This is a complex of very different pathological states we just put in one place, in one pool, but in fact they're all different. It is like in old medicine, they try to cure all diseases by letting blood out, just by making some, or by giving you some powder that causes the diarrhea. So they just put all different diseases in one pool. This is maybe what happens with Alzheimer's. So what we call Alzheimer's is a complex of very different diseases having totally different genetic and proteinous mechanisms. This is one idea. Second idea or second information I want to share with you, there is a good, interesting book written by medical doctor Dale Bredesen. I will write his name here, Dale Bredesen, I think like this. He has introduced an interesting program which is called RECODE. RECODE program. Please take a look, especially if you have parents who are in the age, or maybe your grandparents, and you may contribute to keep your parents or grandparents healthy, or maybe also it would be true for you as well. you may take a look. It's an interesting things. You can find his books, his videos, his papers in the internet. And I found this interesting because they studied Alzheimer's disease for around 20 years, and they established at least 3 pathological mechanisms of Alzheimer's disease. So they are talking about inflammatory nature, they are talking about genetic nature, and they are talking about some, yeah, I've forgotten how it's called, but third mechanism, spontaneous nature. They argue that Alzheimer's disease appears primarily as response of the brain to some inflammatory or toxic process. So they say the core thing is intoxication. So it is the response of our brain to intoxication. And basing on this assumption, they developed a program consisting of 36 steps, how you can not only prevent Alzheimer's disease formation, but also counteract. So what they report, I don't know if I should believe or not, but seemingly yes, they reported about patients that already had initial stages of Alzheimer's disease and then entered the record program, and they recovered, they reversed Alzheimer's disease changes. Sounds fantastical, sounds interesting, and I recommend you to take a look and to discover what they recommend. Very briefly, it is actually nothing special. They argued that we should not drink alcohol, we should not smoke, we should pay attention what we eat, how we sleep, how we expose our brain to the light. So those things. And they just ask or they say that those things slowly can intoxicate our brain. And well, to me, sounds quite interesting. Not necessarily that it's totally true, but at least nice and interesting thing, nice to know, you know. By the way, it's not just like a dilettante point of view. He's a medical doctor, and he studied the exact mechanisms of Alzheimer's disease for many years, and he found the role of neurotrophins in this. And here, as a molecular biologist and biophysicist, I totally agree. Obviously, neurotrophins, hormones that guide neuronal growth, they do contribute in Alzheimer's disease, and he was one of the first persons who noticed this neurotrophin mechanism in Alzheimer's. Okay, sorry for telling a lot about this. To me, it's an interesting thematic, and I hope for many of you in Research Center Jülich, there are many research groups who work on Alzheimer's disease mechanisms, and slowly our students are also joining these groups. Every year, I have 3-5 students who work on these topics, and that's why I wanted to share it with you. And maybe you will participate. Sorry, I spelled his name wrongly. Bredesen. Yeah, so I think like this. Bredesen, not Bedesen. Bredesen. So, any questions so far? I know it's we make it relatively quick, so you probably don't have time to write your questions, to write comments. So that's why I'm also making some breaks. Fine. After we've spoken about tau protein and its role in its possible role in Alzheimer's pathology and its function as microtubule stabilizer, let us address one more medically relevant issue related to cancer treatment. And cancer is yet another absolute hot topic nowadays. Just to give you one number, nowadays everybody talking about corona, but there are 10 times more people dying, or 100 times more people dying because of cancer daily. And this is something that in many cases we cannot help. For some cancer types, it works already. There are vaccines, there are drugs, but 90% of cancer types remain really uncurable unless you make surgical operation. That's why cancer remains so important. And now let me tell what cancer has to do with microtubules. For that, we need to come back to the origin of microtubules. As you remember, most of them appear in the cell or start their life from the microtubule organizing center. In some books, it's called centrosome, right? So centrosomal structures. In some cases, in botany, they call it centriole. But in fact, the good name for that will be microtubule organizing center. It is a complex of different proteins and containing tubulins as well. So special type of tubulin, so-called gamma tubulin, which serves as a seed for the growth of microtubules. You don't need to know about fine composition of it, but we know that such structure is necessary in a cell, and many microtubules, they start growth there. Now let's think about what happens as the cell divides. First of all, DNA replicates. So we have one cell, but two DNA copies in the cell. And now we need slowly to think about separating daughter cells and cellular division. For that, you need that every cell will later have a microtubule organizing center, because otherwise, one of the daughter cells will not have it. In order to make sure that every daughter cell will get microtubule organizing center, it needs to be duplicated, right? It will be duplicated. And it really happens before cell divide. And then we have now two centers where microtubules originate. And now from these centers, we'll have microtubules that would catch chromosomes. And what we observe them is a movement of chromosomes, this mitotic spindle, right? Okay, let's take some figures about this. These are two, so this is centrosome. It's a single centrosome without division. And these are some pictures of microtubules. You see, most of microtubules, they originate from the cytosome. And there are also some microphotographs showing cytosomes, showing the microtubule organizing center to you. So again, I want to repeat, centrosome and MTOC is the same. It's just different names. So this is a single copy here. And here we see before cell divides, also its MTOC needs to divide. It will be duplicated. And then every this center will serve as center for chromosome pulling and pushing. So this is just, again, one more time, this MTOC, fine structure. So here we see microtubules attached. So actually plus ends are showing outside, and minus ends are attached to the MTOC. Let us think for a second. Why it is that minus show inside and plus showing outside? I think it's quite logical because, you know, plus end of microtubules grows faster. Yes, it is about growing. And that's why, of course, it would be kind of stupid to aim plant ends inside because they have no space to grow. So usually the orientation is plus to outside, so this more fastly growing end will have space to grow, can perform its function, right? And now we are coming to the cancer treatment issue. Look, what we know about. So there was a question about what's the minus end is binding. So it's binding to this gamma tubulin, to this core structure, to this core I showed you. So here we see again plus end and minus end is binding to the, well, it's a complex of proteins. So mainly it is gamma tubulin, but there are a couple of more proteins that make it, so it's a kind of anchoring there. So the minus end is anchored. It's like blocked there and cannot grow, of course. Let us now consider what is cancer. In this course, we'll see that cancer is not always caused by genes. It can be caused by extracellular matrix. It can be caused by adhesion, and it can be caused by misfunctioning cytoskeleton as well. But what is cancer? We have uncontrolled cell growth, right? So some cells that start uncontrolled proliferation, endless division, and they show aggressive behavior. They grow, and then they start spreading over the body, causing metastasis. But usually, as the cell undergoes malignant transformation, there are many mechanisms that will kill the cell. And in 99% of cases, it works properly. Now you are sitting in front of your monitors and me. Think about this. Now, at the moment we are talking, a couple of your cells were underwent this malignant transformation. It happens. It's true. And in my body as well, now 2 of my cells maybe decided to be cancerous. But luckily, either they will kill themselves in a process called apoptosis, or they will be killed by our immunity, right? And one of the mechanisms that takes care that such malignant cells cannot proliferate, cannot divide, is that they mostly have multiple microtubule organizing centers. Let me show what I mean. So the upper row shows, this is, by the way, our master project. One of our master students did it in Forschungszentrum Jülich, and I supervised this work. I found it really fascinating. And if you are interested, you can also find similar projects. It can be done either in the Uniklinikum in Köln, in Cologne, or in Jülich. So there are lots of opportunities. There are many groups in Jülich and Aachen and Köln who work on cancer schematics, by the way. So let's see the healthy cell, the normal cell. So it has one single microtubule organizing center. And as it divides, the chromosomes are properly arranged. The blue bodies are chromosomes. And as the cell divides, the chromosomes are evenly distributed between daughter cells. Even the cancer cell line, those cells are, damaged. And I should say, luckily, mostly these cells, they possess different or several microtubule organizing centers. And as such transformed cell tries to divide, what happens is every microtubule organizing center tells me, I will take it. So, and they all pulling microsomes in their direction. Now I see that the video, in my case, is slowing down, so I hope that you still can hear me. I want to show it here. Look, if you look closer, this is what I described. There are several green microtubule organizing centers, and every of it is pulling microsomes to its direction. You know what will be the consequence of this? I think you can guess, right? Such a cell, after it divides, will have unequal amount of chromosomes, and both cells, both daughter cells, will be not viable. They both will die. And this is, of course, a great thing. That means also passively, our body has mechanisms that remove cancer cells from body. So they divide and they die after that. But now a bad news is coming, and this bad news is that cancer cells in our body, some of them have learned to group, to cluster cytosomes. That means if such a cell has 5 cytosomes, it clusters them into groups, and then if every group works as a whole, as one team. And now they are pulling chromosomes in a very coordinated way so that every cancer cell gets equal amounts of chromosomes again. So this is kind of next step in this weapon race. So cancer cells, some of them are capable of phenomenon called microtubule organizing center clustering, or centrosome clustering. So here, some questions came about, can we prevent cancer? And again, the PowerPoint crashed. Sorry for that. I will start it again. Meanwhile, I can answer your question. Well, in fact, in some cases, we can say we are preventing cancer every second. Our body prevents cancer. Our body prevents cancer in that it exercises its hundreds of natural protection mechanisms. So for cancer to appear, multiple events need to happen. So multiple mutations need to happen. And our body must fail many times for cancer to appear. From this point of view, one thing we can do to prevent cancer is to take care that our protection mechanisms function. So now I will relaunch it again. Just a moment. And after I will tell about this, we will have our next break. So this, and now let me share, right? And now I only need to drag my chat windows. And participant windows to a separate folder. So just 5 minutes, then we have our break, right? But I just want to finalize this part. So I hope I could answer a little bit your question, can we prevent cancer? Well, yes and not. Uh, Maybe I have good news for you, all of you actually. Before medical doctors and most of people knew that cancer is deathful in many cases, and without treatment, it will lead to your death. Recent studies and the evidence-based medicine shows that this is not true. And in fact, it is now known that if you consider very old people, like people who die with 80, like later than 80 years old, 90 years old, they die because of some other reasons, because of cardiovascular diseases, because of pneumonia, because of something else. But if you pathologanatoms, as they open their body, they find quite often that those people had cancer. They had, in case of males, 80% of old male persons have prostata cancer. But they just don't notice this. It is there, but it is so slowly proliferating or self-limiting process that in about 80% of cases, cancer remains unnoticed, and it's not what causes the death. I hope it's a good news for all of us. And by the way, that's why there is an interesting reasoning I read shortly. It is not good to make a total screening for cancer, for some types of cancer. For some, yes, but for some it is disadvantageous because if you test 10,000 people, in 5% of cases it will be false positive. And that means you start treatment, but these persons, or chemotherapy treatment, but it is a false positive. So the people will suffer, but totally in vain. They have no cancer, but you are treating them. Second, also in 20% of cases, or even more, depending on cancer type, up to 80, this person will never notice that he or she has cancer. Maybe you see there is an interesting argumentation that in some cases screening for some cancer types is obsolete. And by the way, this has been done before for uterus cancer. They suggested screening, but now they said no, it's not. This is not a deathful cancer. If it occurs, it can be handled. And if not, that we better do not do any screening. So you see, it's an interesting ethical question, and modern medicine came to conclusion that screening and early diagnostic is not always good. Funny, right? So quite strange, but it is like this. So here's another question. Taking in mind the chromosomes clustering, will they speed up the mitosis cell division? Oh, good question. I don't know, honestly. I've never checked it in terms of speed. To my knowledge, possibly not, because cell division is a process that contains many contributors, and if microtubules work faster, it doesn't mean that actin that serves for cytoplasm division, it can follow same pace. That means I do believe, it's just my guess, that cell division is not really accelerated because of that. Maybe I'm wrong, but this is my guess. What our master students have found is here what you see on this graph. And this is that what you see here, just a moment. I need to a second. uh, laser pointer. Yeah. So what you see here are different cell lines, and those are cancer cell lines that has been examined. And what I checked is the number of ciliated cells in between this among these cell lines. So only the first cell types, these are normal human cells. Interesting picture, right? Seemingly, cancerous transformation greatly correlates with the presence of cellular structures in the cell. So somehow the cellular organizing center is affected, or in some cases blocked or clustered in the malignant cell types. What I'm telling about is this structure, which many cells possess, is like primordial cilium, is a seed of cilium, and interestingly, some cancer cell types, they transformed it in a way so it remains undetectable. Let me explain now the whole picture. First, normally, cancerous cells die because of these multiple centrosomes. Some of them have learned to cluster their cytosomes, and then they can survive and proliferate in the body and divide successfully. What we can do now, there are drugs, there are substances that prevent centrosome clustering. So one possible therapy of cancer can be that we prevent centrosome clustering. In other words, we disturb cytosome clustering in the cancer cells so that they experience this previous situation. They have too many cytosomes, they cannot divide properly, and they die. So, and this is one of the interesting and prospective directions in cancer research, so that we aim at microtubular structures. We aim on the structure, fine structure of microtubule organizing center, and by doing so, we try to we try to disturb centrosome clustering. So this is one of the promising strategies in cancer treatment. So I hope I could tell you something interesting and new concerning microtubules, cancer, Alzheimer's disease, and now you have some sort of picture about microtubule roles functioning in pathology and physiology. Now let's make a break, and 10 minutes long, and so that you have power for our last section, where we move towards motor proteins, kinesin dyneins, and where we talk about intermediate filaments. Let's see how much time we'll have, how quickly we manage it. Maybe that will be it for today. No stress, everything's fine. I hope it was not so quick, not so slow. So we see us in 10 minutes, okay? So I think we are back, and for our last session for today, I promise it won't be very stressy for you because most important things we've already discussed. So it won't be much. First, let me. Now we are finalizing. I'm giving last remarks about microtubules. We are finalizing this chapter, and in order to proceed to motors and intermediate filaments, to finalize, let me mention one more fascinating and mysterious aspect of microtubule functioning. And for that, let us think about general anesthetics. I'm talking about not local anesthetics like lidocaine, but rather those substances that are used for operational narcosis, right? So I think narcotics would be a wrong term, but I think you understand what I mean. So substances we let our patient to breathe in, and the patient will be unconsciousness, and it will. You can make surgical operation, and the patient will not feel pain and will not see anything. So there are many substances like these. So historically, most famous were laughing gas, N2O, ether, chloroform, but they all turned to be dangerous. Later, some organic compounds, other organic compounds have been developed. But what still remains mysterious is why do they act, and how do they act? What is in the substances what makes people do things? So they lose consciousness and they not feel pain anymore. And the first hypothesis was that it's related to biological membranes. So those substances act on membrane channels and they just do something on membranes. That was logical, right? But later it turned out that it's not explaining all phenomena. Maybe some of you know that even breathing pure argon or pure krypton, well, not pure, mixed with oxygen, will make you unconscious very quickly. So you will just fall down, and for 20 minutes you will be off completely. And you will feel no pain. So that means if somebody cuts your leg away, you will feel nothing. So from this point of view, things like argon and krypton, they are perfect general anesthetics for narcotizing patients because they have no side effects. They act quickly, but nobody knows why they work. Just pure argon and oxygen mixture, how comes? Argon is an inert gas, the krypton is an inert gas, noble gas. How do they work? That's why many people thought about this, and maybe you know some names like I think Stephen Hawking tells you something, right? Stephen Hawking, his friend Roger Penrose is another famous physicist and mathematician. And yet, next person in this club is called Stuart Hameroff. So this is like a club of 3 people. Stephen Hawking, you know, for his ideas. Unfortunately, he died a while ago, but his brilliant mind, in spite of his disease, really fascinating scientific life, But his both friends, Roger Penrose and Stuart Timirov, are still alive and continuing his developing his ideas. So one of them, Stuart Timirov, and here's the next slide, has noticed one interesting phenomenon. He noticed that all substances that cause this narcotizing action, these two effects, loss of consciousness and loss of pain sensing, they all destroy microtubules, or at least they all affect microtubules in the cell. So, and this led him to hypothesize that microtubules are structures in our body that are responsible for consciousness. And well, sounds quite crazy, but you know, many ideas in science, they were first crazy and nobody could believe them. One crazy idea from Copernicus, one crazy idea from Darwin, one crazy idea from Einstein. And here's yet one more crazy idea that might come true, and this is that microtubules in our neurons additionally serve as consciousness material basis. And I, from my side, know no argumentation that is against his hypothesis. Moreover, our knowledge about post-translational modifications of microtubules, and this is what you see here, suggests that there is a hidden function those structures perform. There is like domain-like record cluster structure, some code written on microtubules, what we still don't understand. So I know it sounds too crazy and well, but still some of you who feel interested and you want to know more about this, Stuart Timirov published a book. It's called Quantum Consciousness. Or you can just search for in the internet. There's also a web page of him together with Roger Penrose and Stephen Hawking. They discussed a lot about these issues, and these three brilliant minds of our recent science, they all agree that might be true about this. So they argue that microtubules are where our consciousness, our self, perception, and our memories are stored. Who knows? Maybe it is true. So that was it about microtubules. So we finalized, and now our next little chapter. It won't make too much time, and it will be quite simple. It's about motility and about molecular motors. And we already mentioned these two names, two proteins that you please need to know for the exam. Molecular motors associated with microtubules are called dynein, and it moves towards negative direction, and kinesin that moves in a positive direction. That means to the plus end of microtubules. Yes, so here we have the slide which illustrates it. Maybe you don't have it in your script because it's dark and I didn't want you to print it, but there are lots of additional slides you have, and in fact it's a lot of repetition here. So what you see here, these small things are dynein and kinesin. As you can see, they are also symmetric double structures. It looks like they have 2 feet, and they can work along a microtubule in certain direction. So the preferable direction for dynein is towards the minus end, and for kinesin towards plus end of microtubules, so that cell can transport substances in both directions. For example, let's consider a neuron inside of axon, which is very long, and we have a microtubules, and on the one side of microtubule, there will be kinesin molecules moving in this direction and dynein molecules moving in this direction, and they do not disturb each other. This is exactly how we have it on the highway, one side, another side, and the cars moving in opposite direction. This is the picture we have in our cells. This is how it works. So here I'm showing now a couple of more pictures that are just illustrations. Don't worry if you don't have some of them in your script. These are just selected pictures I wanted to give to you to illustrate, to put more input into your visual brain. Here's again dynein as a main molecular motor associated with microtubules. And this is a fine structure of the machinery. For example, in this case, we see how it can be connected to red blood cell membrane. For example, here we have actin-like and spectrin. And also, we see that those structures can be strongly involved in endocytosis and exocytosis processes. So again, what we know as membrane-phatic processes, endocytosis, exocytosis, lysosomes, all this stuff, they do not move passively, you know, like bubbles in a liquid. That would be stupid for a cell. But in most molecular biology or biochemistry books, they just say, okay, these are bubbles, and they just move, and that's it. But now we know at this level, we now know that those bubbles, they are moving in a controlled way. So they are driven, actively driven by molecular motors. And one of the principal players here is dynein, that serves as the really motor, as a pushing force that can transport microvesicles like endosomes, exosomes. Exosomes are excreted vesicles in the cell. So here's a picture. Here you can see this kinesin motion. Something similar we also saw for treadmilling, right? It's a little bit like, looks a little bit alike, but this is a kinesin motility. And in our case, remember, kinesin moves along microtubule. Right? Here are some biophysical additional information that you don't need to learn by heart, but might be interesting for you as reference values. We obviously know that for making force, ATP energy somehow is used, and typical force they apply is a piconewton. No, typical work that is done upon hydrolysis of one ATP molecule is estimated as 80 piconewton multiplied by nanometer. So like it's not what is product of pico in nano. It's so I don't know, femto or attojoules, right? So energy units. And here we are talking about typical forces in the range of 10 piconewton. And indeed, in this course later we'll see this is characteristic magnitude of many forces cells exert or molecules can exert. Is a piconewton range. These are typical things. I mentioned in the cell biophysics 1 already that I am not a big fan of newtons because it is a wrong unit for forces. We better measure energy first as energy in kilojoule. But if we really want to apply newtons, then we are in this range, so piconewton range. And one more thing that is quite fascinating is that this high efficiency, high efficacy of these structures. They said to be 50 to 70% efficient in terms of energy usage. Just compare it with our solar panels, with our heat engines, car engines, combustion engines. With any human-made process, you see that 50 to 70% is really great. So it's really great efficiency. Only 20%, 20 to 30% usually lost as heat. The rest is useful work. Now, one next station for us will be, we need, it's not cell biology course, so it's biophysics course. So that's why we necessarily should have some biophysical issues. And that's why I feel like obliged to discuss at least one theoretical concept with you, biophysical concept, which is called, we are going to understand and revise Brownian-Rachet models. So, Rachet models. I know it's maybe not so, so attractive for you, but I still hope that you will appreciate some beauty of this biophysical topic. We will talk now about some very fundamental mechanism, how motion occurs in the cell, how molecular motors apply this force along the thread. So how do they do it? What's the mechanism? Because you see, we have a problem. They say ATP energy is used, and it's used to make force. Say, okay, fine. But what if you start to think deeper? Say, okay, but how does it happen? What happens there? It is like saying car consumes gasoline and can move. You say, okay, fine, cars uses electricity or some benzene and they can move, alles klar. But what if you just take a look inside of the engine and then you understand how it occurs, right? And this is what we're going to do now, to try to take a look into Brownian ratios. So as we mentioned before, two principal motor proteins that attach to microtubules are dyneins and kinesins, right? And so far, there were two classes of models proposed to explain how they work. The formulas, I know they don't look nice to you, and they look a bit ugly, maybe, for you. But I hope after I explain them, you will see that it's not too bad. So one more time. There are basically two ideas, two classes of hypotheses that explain how molecular motors make motion, how they generate motion. The first hypothesis are they're called directed hypotheses. There is also another name, walking hypothesis. Walking not from work, but walk. And those hypotheses are most easy to understand. It's just about that ATP hydrolysis is used to make conformational changes in the proteins so that they make some like walking step by step walking along a protein, along microtubule. So this model does not explain too much, but at least it is an attempt to see in more details what's going on. So they say about this conformational changes of single myosin, kinesin, or dynein subunits. I think that's clear. Another group of hypotheses, they are called Brownian ratchet models. They are less intuitive, and let me explain how they work. And this is what those equations tell you. What's stated here, they say, well, the motors, they work actually by diffusion. They're called diffusional models, Brownian diffusion models. And they say, well, in fact, there is no direction. The force that is applied is random force, and if we average it over time, it will be zero. So it's just diffusion. But then they say, wait a moment, but how comes that we have directionality here? So if the force is averaged in time, and we're talking about force here, which is averaged in time, how can random force cause directed motion? So biophysical question. I hope some of you are still with me. I'm almost ready. And now the core idea is coming. The core of the ratchet models is that ATP makes participating structures asymmetrical. So it makes asymmetry in the whole setup so that random process results in directed effect. If it sounds unclear, let me show one thing to you, and then you will understand it. So what we see here is, moment. So this drawing is actually the core idea of the Brownian ratchet models. Look, if we assume that here we have some random process and ATP is not used, we have just diffusion, then the subject will move with equal probability to the left and to the right. There is no directionality, and there is no work, no useful work you can do. But look what happens if ATP makes certain asymmetry in the system. And this asymmetry can be visualized in such a ratchet pattern. So look now, if we have this structure, now we make it move randomly. But look what happens. Your setup is limit its freedom, so it cannot actually move to the left, because there's an obstacle there. So it moves in all directions equally, but to the left it cannot move because there is an obstacle there. But it's just vibrating, and what happens, next step, it will land here, and again it's still vibrating, but now it is one step to the right, and if it's continuous vibrating, it will move step by step towards right. And this is what the many mathematical descriptions actually mean with ratchet models. ATP somehow makes molecules to act in this asymmetrical way. It gives directionality to some intrinsically random and isotropic process. So in this case, ADP is contributing to asymmetry. It produces asymmetry. Thank you for your question, right? So the question was, is asymmetry built or destroyed by ATP? I would say if the models mean it's built. So there's another question: to structural changing results in enhancing of potential energy? Well, yes, and this is where ATP energy is really used. So ATP causes structural changes in the molecules and in the surrounding water. It has some different potential energy now, and this structural change is used to give directionality, to make directed motion now. That's not a very easy, straightforward process. It's hard to understand, and nobody knows all the details. But this is the current state of art, how we understand the participants of ATP in molecular motor processes. Here I should call another name or mention another name, Richard Feynman, who first proposed perpetuum mobile, so the engine that would work on diffusion. If you want, you can check in the internet, Smoluchowski, Feynman, Richard Feynman, Nobel Prize winner. He proposed such a ratchet model. He says that if you have gas molecules that move randomly, they will push the wheel in all directions equally, so it will vibrate. But because of the special asymmetry in the wheel, because of its peculiar shape, it will preferably move in this direction. And this is what he announced, and he asked people, okay, explain me why it will not work, because it would be engine that works without energy, without any fuel, just by using diffusion. So it would be this perpetuum mobile. In reality, such an engine has been even built, but it only works for very big particles, and it has been shown that on microscale, this engine will not work because also the axis of the wheel is also subjected to Brownian motion, so that's why the wheel will lose directionality because it will slip, it will skid to the opposite direction as well. So many people have tried to build it, and actually in the end they showed that thermodynamics is still true, that perpetuum mobile is not possible. But if you have ATP, if you make some gradient, like thermal gradient, such engine will work. And this was a nice triumph of thermodynamics. They showed that yes, such structure works, but only if you either consume energy of chemical substance, or if you make a gradient here. So there must be gradient, must be an equilibrium on both sides of the wheel. So there is a question about so ATP acts an obstacle. Not exactly, but it causes protein to change its shape, and we don't know how, right? I'm just explaining the current mathematical models. This causes directionality. This causes our whole molecular setup to act like such ratchets, such this zigzag structure. And this is what ADP is doing. Maybe ADP serves for stabilization of the structure in the vertical direction, so it will not slip. Or maybe ATP is that what makes it obstacle. That's also possible, in fact, right? Nobody knows exactly. So another question was, so the conformational changes are in microtubules and random motion occurs with a motion protein. Yeah, so this model says that taken alone, microtubules and molecular motors, they are kind of symmetrical. If you put them together, they will interact and they will move, but they will move to the left or right with same probability, so nothing interesting will happen. So that means this diffusional model says molecular motors work without ATP, no problem. But they miss directionality. They just cannot decide in which direction they go. Addition of ATP or GDP in some cases helps them to decide, gives this direction, says, okay, hi, guys, you need to go there. How exactly it works, we don't know. Thank you for many questions. I haven't expected this because mostly at this point of time, most of the people are a bit tired and kind of a little bit skip this. And that's why I don't want to really focus on it so much, but still to me, it was important to mention these interesting ideas. And I think if I mention at least this idea of Brownian ratchet, you have an idea about this because we met it already 2 times. We met Brownian ratchet idea in case of pushing actin motion, and again, it was like a random process actually, but because of actin polymerization guided by ATP, there was a pushing force applied by actin on the membrane. And now we met it again, somewhat updated and revised, where we again acquainted some mechanistic idea and mathematical description of those phenomena. For some of you who are interested in physics and mathematics, you can take a look. These are potential equations. But people who are not so great fans of physical models, you can skip it. Nothing will happen. I am not going to ask anything about mathematical description potential, not in these relationships at least. Yeah, so Caroline mentioned that it's a little bit hard to understand and to relate to the equation. And actually, that's why I try to make it different. Because what the equation tells us is that there are 2 factors acting and produce directional motion. So what we see in this equation, it is a force, which is a function of time, but this force is fluctuating. It's totally random. And this is just an agitation which serves as the actual source of motion. And there is also a potential term, W, which is where ATP is contributing. So ATP changes the potential slope, energy slope of our setup such a way that this energy slope generates directionality. So what I tried to do is to explain this indeed tricky formulas in more human language, you know, in more biological and easy language. Anyway, I hope it worked at least to a certain extent, and if not, it's also not too bad. Still, let us now go ahead and we see some other pictures here. Again, kinesin, dynein, tiny workers, hidden heroes that many people are not aware of, but the most transportation job in the cell is done not by simply diffusion, but by cytoskeleton accompanied by dynein and kinesin. So now you know that processes like endosome formation, so pinocytosis, endocytosis, exocytosis, they are all occur with participation of motors. In synaptic cleft, by the way, there is another class of proteins, motor proteins, septins. Not septins, I've forgotten the name, sorry. Okay, but at least here also nothing happens spontaneously in a synapses. Also, there are motor proteins that transport vesicles to the synaptic. Yeah, thank you. So here came another question. What does it mean, intermediate filaments, EF? And this is where we are slowly coming to the transition from microtubules to intermediate filaments. So we are leaving now slowly over molecular motors. We are leaving microtubules, that was it, and now we are entering the intermediate filament field. And in reality, it has been shown that molecular motors is not only about microtubules, but also it has been shown that intermediate filaments also are involved here, and that means all three types of cytoskeleton, actin, microtubuli, tubuli, and intermediate filaments, they are all connected in some very dynamic, very actively functioning physical network. So here's one more picture about this, where we see that all three different types of cytoskeleton are actively interacting to each other in the cell. So they're not working separately, but there is a synergy. So we have microtubules as main highways of the cell here. So there's dynein kinesin moving along. Here there is myosin, protein, and actin. And also you see there is also connections between those components and intermediate filaments. Let me skip this slide because I've spoken a lot today about cancer, and that's why I will just show some couple of more pictures about proteins. These are walking models, not diffusional models, but walking models. And well, nobody knows how it happens, but it could be like this, or maybe like this, or maybe like this. If such a model is true, at least. And so I'm going further because it's not so important. Here are a couple of more pictures. This is again the whole picture of how transportation in cell works. So here you see a neuronal cell, a nucleus, and substances need to be transported to the synaptic, to the terminal here, and they will also need to go backwards. So for the forward motion from the nucleus to the periphery, mainly kinesin is doing this, and on the way backwards, there will be dynein that will be the transportation vehicle. That will be the driving force. And this yellow circuit is just a cargo. It's what's transported, cargo, right? So the luggage. So some nice pictures about microtubule involvement in chromosome division. You see, we are finalizing this topic. We are leaving this topic now, and just to finalize, I'm showing some pictures to you. Nicely labeled microtubules, perfectly arranged. By the way, you also please notice that every single chromosome is held not by one microtubule, but multiple. So it means if something wrong happens, there are many ropes that support chromosome. So usually it functions quite good in a cell. And one more thing I want to mention is that microtubules can not only push but also can pull chromosomes. And this is what makes additional safety to this process. So our chromosome division relies not only pulling but pushing as well. And here it is good visible that from the both sides chromosome move. That means if something wrong goes here due to some mutation or disease, the other side will still do this job, and it makes sure that our cells will divide properly. So such, smart machinery. And here are our respiratory cells here. This beating motion we can see, and the fine structure of these cilial structures that mainly composed of dynein and tubulin. Now I must tell you, I always have to fight with myself, because there are here also lots of things from biology and medicine I want to tell you, but mostly it's just too much, right? So also here I could tell you about how conservative this structure is through the animal world and plant world. So this design of this multiple wheel design is extremely conservative in nature. So in our cells, we see same shape, and the same you will see in plants, the same you'll see in infusoria, in whatever, insects, whatever. Everywhere we have same design. So seemingly nature has once found this mechanical design, and it worked. And this is the design used for produce ciliar wipe-like motions. Maybe to finalize, why those wipe-like motions are produced? How comes that this type of motion occurs? So how do microtubules make this type of motion? Let me explain very briefly. It's very easy. It happens because you have your microtubules tightly connected first to each other, but they can slide with respect to each other, and they are tightly connected to the background. This is what you see here. So what dynein is doing, it produces sliding motion of two microtubules with respect to each other, and this causes in curvature, in bending. So sliding of microtubules makes them bend on microscale. So I found it interesting, and I wanted to show it to you briefly, how sliding motion can be transformed into bending motion, and this is what we see here. Fine. Sorry for taking much time, but still, a couple of slides I want to force into you. I know that you are tired, but like a couple of minutes more just to make one topic finalized. The thing is that intermediate filaments is a short topic, and it's worthy to do it today. Okay? So that's why we're going now to jump to this very short chapter devoted to intermediate filaments. Here we have the network of intermediate filaments. This is the last component of the cell. And let me just briefly and quickly explain something about them. First of all, these are components that are really rope-like. They are really flexible, and you can always think about them as about ropes. They are ropes, they are threads. And as every rope, intermediate filament is totally perfect in withstanding tensile force tension. It's really great, it's very strong. But it's very bad in pushing. It cannot push. Physically, you cannot have compression in the rope. You only can tension in the rope. And this is absolutely true and mechanically applicable for intermediate filaments. These are ropes that, by using tensile force, gives mechanic stability to our cells. And it is only here where we find this smart engineering design that you don't use these stupid spherical structures to make mechanical rope, but you use elongated long elements to make rope out of them. So you use some chains, some long building blocks, you combine them to make a rope. And this is how ropes made in technique, right? So you take a thin rope, you twist it with another rope, and another rope you twist with another rope, and another rope you twist with another rope, and now we have a nice, strong, big rope, and this is how you do it. Saskia is giving some signals that she's tired. I know. Okay, last slide and never. Okay, just very last slide. And that's actually almost everything what we need to know. Next week we can just refresh it. But basically, intermediate filaments are exactly built this way. So we see tiny ropes that are combined together to produce bigger ropes. And if you think about some intermediate protein that you need to know, I think everyone knows this name. It's keratin. So keratin is, I think, the name that appears in all of us. And we know that keratin is strong. It's about nails, hairs, stability of our skin, and so on. So it is keratin that represents this structure in a very ultimate way. As we'll see next week, there are a couple of more proteins, but there are too many. And good news for us is that you only will memorize these two names, keratin and vimentin, and that will be totally enough. I see, thank you for giving me signals that are getting tight. I hope still that most of you understand that since we meet once in a week, I try to squeeze, to press as much information into you as possible, just to spend our time efficiently. I think you will be able to recover after this class and also maybe after your job. And you forgive me for maybe making it a little bit too intense. So you asked about the second name, right? So let me find it here. Vimentin. All right. Vimentin. So this one. These are only two names that we will talk about them next week then. So today we did a lot. Thank you very much for your attention. I know it was hard. It was a lot. And this is how this course is so far. I still try to make it a little bit amusing for you, just to animating these very dry things with some stories. Because otherwise I just say, okay, you have to learn this and that and this, and you learn it and you come and you pass the exam. But I think it's not such a good idea. So I hope, in addition to learning names, we also experience some practical projects, some current information, and I think it worked in many cases. So have a nice weekend. Enjoy your time, and hopefully the weather will remain sunny and good. Think about your homework. It's nothing special. Just think about filling our table we discussed about. See you next time then. Bye bye. Thank you.